Hello, Assalamu alaikum. Please let me know is I am audible?
Yes, Miss Afshan, you are audible.
Okay, thank you so much, Mansoor, for letting me know. Hello, everyone. Assalamu alaikum, and a very warm welcome to everyone on behalf of Beta Nova International Alliance for Sciences. In our course, Advancements in Molecular Biology 909, here is me, Hafsa Sarfraz, and I am going to host today's session. And today marks the beginning of an exciting learning journey where we will explore cutting edge tools shaping the future of biological research. We are deeply honored to host our distinguished guest speaker, Professor Dr. Yusof Deeni, former Director of Research and Dean, School of Postgraduate Studies, Federal University, Jigawa State, Nigeria. With his remarkable expertise and contributions in the field of molecular sciences, he will be enlightening us on one of the most transformative technologies of our time, CRISPR-Cas9. We extend a warm welcome to Professor Dr. Yusuf Dini and express our gratitude for joining us today. We look forward to an inspiring and insightful session. And students, if you have any queries, you can write in the chat and we will deal with your queries at the end of the session. And no worries about the attendance, it will be marked. Okay. Professor, over to you. You can start now. Thank you so much.
Okay, thank you. Good evening, all. And today, as the introduction did mention, I'm going to speak about CRISPR-Cas system, which is cluster regulator, regular interspace short palindromic repeats. Uh, this is the outline of my presentation. There will be a bit introduction on general about cloning and how we lead into CRISPR system as a system of cloning. And then I will also highlight some aspect of CRISPR system that has been where artificial intelligence has been applied in order to improve the targeting of genome and cloning. Slide number 2, please. Uh, Usually, cloning is aspect of genetic engineering, which we use, but also we can call it even genome editing. And it's this thing happened naturally anyway, but it's just now with advent of technology, molecular biology, that we're able to harness this technology in an artificial way so that we can amend what is there in the genome. uh, we are familiar probably with homologous recombination when it comes to what replication. And this is, in fact, the basis, fundamental basis of how these technologies or tools are utilized. Artificial homologous recombination and gene targeting and editing started way back through the use of a technique called zinc finger or tools called zinc finger nucleases. It is an innovation that allows specific sites cutting of genome with the aid of fork enzyme to create DNA double strand breaks. However, the design and assembly of zinc finger nucleases were complex and their off-target effects quite penetrating to pose limit for their wider application. Because when we say off-target effect, meaning when we are targeting, we miss the target, and then we're hitting at something that we shouldn't be hitting, and that can create a serious fundamental functional problems as far as the genome is concerned. So, next slide. Uh, so after that, then people came up with the idea of another method that improved the zinc finger nucleases, and this is called transcription activator like effector nucleases, TALENs, as a simpler designs with greater sequence recognition, versatility, and or efficiency, and these are are less complex and less time consuming than the zinc finger nucleases. And in fact, they've been used and applied to create a lot of transgenics. Talents have less off-target effect than zinc finger, However, both zinc finger and talon rely on specifically engineered protein domains, which is called the zinc finger or talon repeats, to recognize and bind to specific DNA sequences and target. Then, along the line, the Cre-LoxP system came on as a non-nucleus, added homologous recombination. Unlike the other two I've mentioned, this is a non-nucleus and is a gene targeting and editing tool also. Next slide. CRISPR-LoxP system uses site-specific Cre recombination that specifically recognize and cut at LoxP site of DNA sequences flanked by LoX site to be removed, inverted, or rearranged. So the Cre-Lox system is particularly used to generate conditional gene knockout or gene knock-in. That means to add or to remove certain part of the genome or to even modify through deletion, addition, or gene fusion at a target site in a cell or tissue specific pattern as controlled by the expression of Cre recombinase. Because this is another advantage in the sense that you can use targeted cloning technique to target a particular cell or to target a particular tissue, not a whole systematic system of the organism. That means we can do conditional knocking or knockout in liver cells or in liver tissue or in kidney tissue or in skin tissue. And this is really a very good, remarkable breakthrough in the sense that certain diseases, certain issues are localized to certain tissues or organs. And that means where we are tampering or manipulating is at the precise size of where the problem is and at the site where we want to solve the problem. And that's why this conditional knock-in, knockout becomes very relevant because you can switch on and switch off depending on need for fundamental molecular biology research. So the orientation and location of XP sites determine whether Cre recombination produces a deletion, inversion, or chromosomal translocation. Cre-LoxP is more precise and with less off-target effect than talent. Next slide. So then suddenly came the emergence of cluster regulatory interspace short palindromic repeats as genome editing tool. Why? Because we are still trying to find cheaper and simpler and more versatile tools than zinc finger, nucleases, or talents, or even the LoxP targeting approach. So CRISPR allows easier design. In fact, it provides avenue for multiplexing and is less laborious in creating whatever target we want to do, and is faster and more adaptable tool for high throughput screening of genome editing. So we can do high throughput screening with this technology. CRISPR is more precise, as I've said, with less off-target effect also, and that gives it the advantage above the other three I've mentioned. That is zinc finger nucleases, talent, and then the Cre-LoxP system. CRISPR uses design-guided RNA, which in this case we call gRNA, that guides a nuclease, which is Cas9 in this case, but there are other Cas systems like which I will mention them 12 and 13 later, to a specific target DNA site through hybrid formation and for recognition. And in fact, because you are using the RNA to guide and it's complementary, that's what allows you to be able to target precisely a particular locus within the genome that you want to do the editing. So this gives a summary of what I have just mentioned about the three methods of gene targeting and then coming on to the CRISPR-Cas editing technology. Next slide, please. So the CRISPR represents a family of DNA sequences found in the genomes of prokaryotic organisms, such as bacteria and archaea. A prokaryote, if you remember, is a single cell organism, often bacteria, and archaea that lacks also a nucleus or other membrane-bound organelles. So CRISPR sequences are derived from DNA fragments of bacteriophages, which are viruses that normally infect bacteria that had previously infected bacteria quite often. So they are used to detect and destroy DNA from similar bacteriophages during subsequent infection, meaning bacteria is using this system as a defense against bacteriophages. And so it is more or less seen as an adaptive immune defense system. That's what it is, and that's what we know what it is for long, long time, over 20 years, close to 30 years, even when it was first discovered, before we realized that this is actually a cloning tool that we can adopt and utilize for successful cloning. So, and here the next, I'm just trying to explain what bacteriophages are, diplonovira or polynovarium, viruses that infect and replicate within bacteria and archaea. And if you remember, viruses, what they do when they get into a cell, they what? They arrest or seize over the replication machinery of the host cell in order to replicate itself and then propagate itself within the host. So next slide. Uh, CRISPR-Cas associated the enzyme nuclease that uses CRISPR sequence as a guide to recognize and cleave specific strand of DNA that are complementary to the CRISPR system. This makes script system to be simple, rapid design by mere creation, and a change of a short RNA sequence, which we call the guide RNA or single guide RNA sometime, containing a CRISPR RNA component, and then a part of it also within the system is a transactivating CRISPR RNA region. And it is this region that actually activate the nuclease activity, without which you can. So within the so-called SG RNA has a proto-spacer motif, which we call PAM, and each cas, that is the nuclease, 9 in this case that I'm using, has specific PAM sequence to target. Next slide, please. So the crRNA provides or contains the target specific gene complementary sequence, while the transactivating RNA is scaffold for binding the crRNA to the Cas9 nuclease, forming a functional complex that guide the Cas9 to the specific site in the target DNA for editing. So the crRNA component of the so-called guided RNA molecule contain 17 to 20 base pair sequence complementary to the target gene. And that's what makes it more specific and more precise and very clean in the targeting, because if you remember, it is only dimerization in DNA. You have complementary pair. And that's how this system is able to guide and locate exactly the location we want to effect the change or the cloning that we want to do through this complementary association of the sequence that is complementary to the target DNA sequence. So the CRISPR system is ideal for gene knockout or knock-in, regulating gene expression and function, multiplexing, and also for high throughput screening. Next slide, please. So mapping, this slide is just demonstrating a cartoonist way of understanding the concept. You can see how the DNA, you denature DNA, you open it, you create the block in terms of replication, and then this guided RNA is able to find access into that particular palm space, and then is able to bind to those complementary sequences. And from there, the system knows that that's the location where we want to effect the change in terms of the guiding. And it is through that process that you can either cut and create a space, and then within the space, you either seal up the system, that means you are now excising and removing, you are knocking out, that in the case of knocking out the gene or the function, and then in the same space, if you like, you may insert something that you want to include as a new functionality. So that means in disease state, you can use this system to excise a default gene and then also use the same system to replace it with what? A functional working gene. And that's why this system has found effectiveness later. I'm going to highlight where it's now applied in even gene therapy. So essentially, this is further cartoonist demonstration of what it is. But in fact, if you remember your DNA repair system. If you remember that, then we know what we call homologous recombination and also non-homologous end joining recombination. So these are the two methods that the technology exploited, and then this method is used to either delete. Usually deletion involves the non-homologous end joining recombination, and then when we are inserting, we use the homologous recombination technology approach or functionality in order to insert the genome. So that's what this actually is demonstrating. The other bits of explanation I just gave it to remind those of us of what homologous recombination is, and then what non-homologous recombination is. Next slide, please. So the next slide is just highlighting historically how I see the whole system started, and how also we are able to be where we are today in terms of the historical dimension of the development of the CRISPR technology in terms of gene silencing or gene insertion or activation in order to clone and improve the technology. Next slide. Okay, so this is the slide I just finished explaining. It's just a summary of what I've said about the CRISPR system, Cas9 in particular, where you can do gene silencing, meaning knocking out and removing, or you can do gene insertion, which is mean putting in something in place. And also it just highlight the historical development of CRISPR biotechnology or molecular biology approach, which started, we first discovered probably the system in the 1980s, but then we never realized how effective and how good a tool it is. We only thought it's just a bacterial defense system. But then later, our understanding of the system makes us realize that we can exploit it as a biotechnological tool or a molecular biology tool. Next slide, please. So this is a summary of what Dudna, one of the, in fact, they got the Nobel Prize regarding this. And they highlighted a number of ways in which we can apply this system in terms of gene therapy, which I've mentioned earlier, a number in agriculture where we can improve crops or improve livestock, and then also in synthetic biology, pathway engineering, all that can be employed, and also in reprogramming approach and certain issue, environmental, ecological issues in terms of even managing vector disease in the case of, say, malaria, mosquitoes. This approach can probably be applied in terms of gene knockout in order to disable the parasite, disable the vector in this case, and you can use it to do viral gene disruption within us to modify things. So that's what people are, that's what they propose in about 2014. And I think as we are now, we know that it's now, this has potential of application in medicine, agriculture, and in a number aspect of biotechnology, which is already happening and is around the place and around the corner. So CRISPR's application, as we say in agriculture, in green biotechnology, plant breeding, crop resistance, plant protection, and all this is done in order to improve agricultural output and to protect plants against diseases and to improve yield and production. And the same in medical and pharmaceutical. People are using it to do gene therapy. For example, sickle cell anemia or thalassemia, they are all diseases, genetic diseases, especially here in this part of the world in Africa, where we have sickle cell anemia and has been around and as genetic. There's nothing, people are consigned and resigned to their fate. But now with the advent of this technology, you can remove the default sickle cell anemia gene and then replace it. The gene that causes sickle cell, which is a hemoglobin defect, and then you can replace it with another new gene. In fact, currently there are several hundreds of people that have benefited from this biotechnology or this gene cloning approach, where they've been cured of sickle cell anemia, and for years now they've not reverted to their disease state, and they can now give birth to normal offsprings without passing their defective gene, because that defective gene has been excised and removed. are replaced with a functional one. So in environmental biotechnology, we can also apply I'm on the next slide, please. On the experimental, you can also apply this in terms of environment, biofuels, where we make bioethanol and other clean renewable energy, and we can also apply it in the development of pesticides to protect plants from pests. In the wide biotechnology industry, we can use it for drugs and in food development. So here, why I highlighted the next slide, yes, is to bring to the idea that we shouldn't be thinking that CRISPR technology is only in the advanced part of the world, but in the rest of the world too, wonderful application of this technology is already there. I've just highlighted a few places here in Africa where they're using research to using CRISPR edited cropping in order to improve yield in sorghum and also to protect against the resistance of parasites, wheat parasites, but also application in terms of gene editing to control maize lethal necrosis in Africa, in which is another challenges in places in East Africa and other. And in fact, even in Nigeria, we are aware that the so-called International Institute of Tropical Agriculture, they are also applying this technology in the propagation of roots and tubers and bananas in order to improve their production. So it's not that it's already there, and I think the limitation or the something in terms of is mostly in the advanced part of the world. But research, as you know, is something you develop through collaboration, and all these African things I have highlighted are as a result of collaboration with certain advanced part of the world, and that is possible, and that is the beauty of science, where there is no border control. As scientists, we are free to communicate, to collaborate, to associate, and advance sciences, and that's I think what this platform is actually promoting. Next slide, please. So a CRISPR system has been advanced with the aid also of artificial intelligence. That is, if you remember, we say part of the problem is the targeting. So the precision of the targeting. So people can now use computer aided this an algorithm that is artificial intelligence to improve the targeting so that has been used to optimize the performance of the guided RNA design, to reduce and also to predict the on and off target effects so that you can avoid them. And that has allowed us also to improve the safety of the use of the technology, enhance the precision in editing, and also help to predict editing outcomes so that we can predict even before we go on to set up the experiment. We can use intelligence to predict to us what is possibly going to happen. So artificial intelligence is aiding the development and refining of CRISPR system for cancer therapy, say, for example, by boosting guided RNA design for precision, less off target effect, and reduce production costs, and enhancing the efficacy of chimeric antigen receptor T cell therapy, for example. So this is a good example I've taken from translational medicine, where it's highlighting the use of CRISPR in order to address issues related to cancer. And you can see there are several approaches here that have been highlighted in terms of what you can do in order to enhance the sensitivity of, say, drugs, for example, where here they are highlighting how they can use manipulation of the nuclear erythroid factor 2, NRF2, which is the master regulator of oxidative stress or responses. In any cell system, mostly, this system are highly conserved, and they are part of the limitation of the success of some anti-cancer or even some other therapeutic drugs, not only in cancer, but in other diseases, because it's a global network pathway that you can find in associated with several diseases, not only cancer, but also in diabetes, cardiovascular disease, you name it. So this is just to highlight how CRISPR technology can be used to enhance the gene editing and to target what we need to repair in terms of cancer, but also this is applicable in terms of other things. And it's not only in, say, human or cancer or mammalian system. You can also go to other lower organisms, animals, livestock you can approach in order to improve, as I have highlighted earlier. But the artificial intelligence helps us to predict before we go to experimental stage, and that cuts down the lengthy time of trial and error in order to get what we want. So uh, there are other CRISPR system apart from the Cas9. As I mentioned, there is Cas12, there is Cas13. And Cas12 system recognizes and clips DNA double strand to produce what? Sticky end. Because if you remember, maybe I mismentioned that. Usually the Cas9 system produces a blunt end, whereas here it produces a sticky end. If you know restriction digest, because sometimes there is issue of compatibility of what piece of DNA you can ligate together, whether it is blunt ligation or sticky end ligation. So the CRISPR recognizes and clips RNA strand, which is very wonderful in the sense that that means we may not even necessarily have to be editing directly at the genome level. In fact, we may even be editing at the transcript level. And that is, in fact, probably a better and safer, I think, in the sense that you do not even have to tamper with the genome itself. It can still be producing what it is producing, even if it's producing faulty RNA. But then this Cas13 system would be able to target that misformed RNA, fix it, and then now it can be translated into the proper function, which is another better, neater way of applying the technology and a bit safer than targeting or tampering with the actual host genome. So we have used artificial intelligence to optimize guided RNA design to reduce and also to predict the on and off target effect of RNA targeting CRISPR tools and to enhance precision in gene editing to improve safety and help to predict editing outcomes. So next slide. So the CRISPR system have been advanced with the aid of artificial intelligence. AI algorithm has been used to optimize guided RNA design to reduce off-target effects, I have said, and also to predict editing outcome. So therefore, artificial intelligence is aiding the development of CRISPR system and therapies in diseases like cancer and other things. So CRISPR used with artificial intelligence to predict an off-target activity to enhance precision in gene editing and promote safety. So, what's the take home goodie in general? I think this technology has the potential. That's next slide, please. Uh, CRISPR technology has the potential to render and sustain development by revolutionizing several fields of study, especially those related to genetics and biotechnology in general. Some of the fields that are the most potential in CRISPR include medicine, as I've mentioned. It has the potential to change how we treat genetic diseases. I've highlighted how we use this technology or is being used to treat sickle cell anemia. People are also trying to apply it in terms of other diseases. So in agriculture, the same thing, development of pest-resistant crops to protect them from disease, but also to develop crops and that are able to adapt to environmental stresses like reduced rain. Sometimes we have drought where some of the crops do not mature because the rain season have ended. So with this technology, we can develop varieties that can withstand and can evolve, can develop within shorter span of time of little rainfall. So CRISPR has been used to produce new byproducts such as biofuels or bioplastics and has the potential to be applied in a wider range of applications such as in the production of industrial enzymes to other chemicals because there are a number of enzymes, like lipases, cellulases and other uh, industrial things that are produced which are very important in terms of our utilization. So from ecological point of view, it can be used to protect threatened or endangered species by modifying their genome and to make them more resistant to environmental stress. So, and from basic research perspective, it has opened up a new avenue for basic research in genetics by allowing scientists to more easily study the functions of specific genes and to effect the gene mutation. Because if you remember, there are a number of genes in the genome that we don't even know what they do and what their role is. And with this technology, we can speed up the way of understanding what the role of those genes are, and what they do. So overall, CRISPR has the potential to advance several fields of study by enabling precision and efficient gene editing and creating new opportunities for scientific discovery and innovation. Next slide, please. So these are just references. I have to apologize that I didn't organize them in the order they should come because of time limitation. But also, I would like to apologize to even those who probably I have used their work but have missed, acknowledging from the references list. So with that, I would say I would like to thank yeah, I would like to thank the organizers of this forum for inviting me to share some of the latest molecular biology tools that are in town, and that I hope most of you and most of us have benefited from this interaction. And I thank you for your time and attention, and thank you so much, and I wish you well. Thank you.
Thank you so much, sir, for such an insightful session and defining CRISPR in a detailed way. Guys, if you have any question, now you can write in the chat so sir can answer your questions. Yes, sir, there are some questions in the chat. Should I read them for you?
You say?
Some questions in the chat are there. Should I read?
Yeah.
Okay, here is someone asking. CRISPR has been used to treat sickle cell anemia, but the child died soon. After, so was it because of any side effect of CRISPR or autoimmunity or something else?
Right. So far, I know that it's been because I have said there are a number of patients, hundreds probably by now. At the initial stage, early, maybe about 40 people were using clinical trial. And to the best of my knowledge, it has been proven to be quite safe and effective. And there are very much less, side effects associated with that. But as you say, steady, if you do something, it's something that you keep monitoring and see how far, the use of such technology is, or its effect in terms of long-term consequences. Because this is something that takes time to monitor and to document. So sometimes it takes a while before we know what is exactly happening, which is similar with the case of where we say environmental heavy metals can cause a lot of disease like cancer in human or whatever. But up to today, nobody can prove to you that it has done so. We can only say that based on animal models that we have been able to use in experimental setting. So, and this is the case with even the use of advent of this technology. It's more precise and less risky as the previous forms of technology. Thank you.
Okay, thank you so much, sir. Here is next question. Can CRISPR be used for sex gene alteration?
Come again.
Can we use CRISPR for sex gene alteration?
Yeah, you can use this CRISPR to adjust to alter any somatic this thing. Yeah, but only that maybe in terms of when you say sex, that means you want to alter sex determination at the early stage. So far, maybe people, this is something worth exploring if there maybe people want to do that. But it's still you can target at early stage because it's genome. Once you are able to access it and then effect the changes that you want to effect. But then the problem with the sex, if you remember, is early on, that much later. I mean, early at the beginning that you have all this conjugation and then the segregation that will result in male, female, whatever you have. But I think it's a worthy area to explore and see whether people can do that. But I think there are more other things to worry about in terms of the use of the technology to improve lives. especially from the context of disease, food production, industrial manufacturing, to better our lives and also sustainability in terms of the environment. And I think that will be more a preoccupation, something, rather than to bother or worry ourselves too much about whether it's male, female, or whoever it is. I don't think. I think there are, in my view, that's my personal opinion. Thank you.
Thank you, sir. Here
Thank you
is
so
next.
much. Can you listen me? Is I'm
Uh,
audible?
y- yes, sir, you are audible.
Okay, dear professor, I have a question regarding CRISPR technology. What is the status of CRISPR-Cas9 and CRISPR 12 and 13 in Nigeria side in the field of agriculture, crop improvement, and in health, allied health sciences
Mhm.
in Nigeria and your country?
Well, in Nigeria, I've highlighted one particular where it's been applied in the International Institute for Agriculture, where they're using it to improve tuber crops. But usually, it's the CRISPR-Cas9 system that is in place. That is the original, the first one that has been discovered. I know I'm aware of that, but apart from that, I'm not aware of any other place in Nigeria where the CRISPR-Cas system is being utilized. I'm not aware yet, but maybe some people have already been able, because even the one in IITA, we call it the International Institute for Tropical Agriculture, is through collaboration with some other countries, which is always the same cases I've highlighted in other parts of Africa, in East Africa and South Africa itself. So it's still in Africa, it's a new, as usual, this thing come from advanced technologies, and then it is only through collaboration and through cooperation that you're able to transfer some of this technology or people acquire such knowledge. So with time, I'm sure we may get to a point where, the other caste system are being utilized to improve or whatever that we are trying to improve in Nigeria. And I believe in the other part of the developing world. Thank you.
Uh, Thank you so much, dear professor, for giving a brief about it. Like in future, maybe we make collaborations with and dry lab in integrated life sciences, so we can work on it further. Yeah.
Yep.
So, Miss Hafsa, please have a mic and continue.
Okay, sir.
Any have question, you can.
Okay, sir. Thank you so much. Okay, sir. Here is
Okay.
next question. How is CRISPR-Cas9 technology cheap as compared to other artificial technologies?
Yeah, that's what we say. That's the advantage it gives. It is more precise and faster and cheaper than the other technologies people are using. Because the other technologies are not as precise in targeting what you want to target. There is a lot of off-target effect, and this has a lot of consequences in terms of off-target effect. For example, if I'm targeted the hemoglobin, the sickle hemoglobin, to fix sickle cell anemia, I should be able to precisely target sickle cell hemoglobin and then fix it, remove it, and excise it, and maybe replace it with a functional gene at the precise locus position where the gene is located. Whereas the other technologies are based on just you are not sure where it is going to be in the genome, where it will integrate, and then you are not sure which locus it is going to occupy. And that's what makes this technology very clear, succinct, and clean in terms of precision. Because the other off target, you see, if for those of us who are familiar with gene expression, if I have a gene and I slot it into any location that it's not its natural locus, then that means even that gene, its expression is going to change fundamentally because I've now been able to move it to a position where it's not its natural position. Because if you remember, there's what you call gene-gene interaction. So the fact that somebody is now in a space where it is not its normal space, it has now found new neighbors, and those neighbors are going to influence its functionality because of the labeliness. And as a result of that, then it may be for good, and in fact, quite often, off targeting is actually a worse situation. And it's very risky because you could, in the process, knock out the function of a normal gene as a result of slotting in something that is not supposed to be there. Whereas with the CRISPR system technology, you are precisely at the locus where the gene is, and you are not tampering with the locus of any other gene. And that's what makes it a very beautiful technology. Thank you.
Thank you, sir. Here is the next question. Can CRISPR be done on eukaryotes as successfully on prokaryotes?
Can come again, please?
Okay, someone is asking, can we you done CRISPR-Cas on eukaryotes as we had successfully done on prokaryotes?
Yeah, it's possible. It's possible. See, the problem with science is this. You can hypothesize, you can think of something, okay? But it is only when you attempt to do it and know whether it can be applied or not, then it becomes a successful tool, or maybe it becomes a challenging tool. So as we use the technology, in as much as we know it is improved and precise and good, it still have some shortcomings. It's not a perfect thing. It's not 100% perfect. We're not saying that, but we're saying that relative to what we have had before, this is a much safer and more precise way of doing the gene editing and gene targeting. And for that reason, people will keep using as time goes on to use the technology. But also, as we use technology, as we know in science, the more you know, the more you use, the more you also find out what is good, but also what is disadvantageous or what is challenging in terms of the use of the technology. I'm not saying there are no challenges, there are, but then it is also in the process of practicing and utilization of this technology that we will know what are the challenges, and then we can be able to overcome some of the challenges. For example, I have highlighted the use of artificial intelligence, which helps us to even use a model where before doing the experiment, you can predict, you know, as in silico sort of experimentation or prediction or decision making that will tell you that if you are to do this, probably it will give you better editing and precision in terms of targeting. And that's how what we have used now to use artificial intelligence also to improve the use of the technology. But then even the artificial intelligence itself, it depends on what component of artificial intelligence are we trying to incorporate, put together with CRISPR system in order to improve the system. Because as I speak, even in protein engineering, to engineer a protein, you have to predict what you think the structure is going to be, what would be its function, which we want to manipulate. Well, with the artificial intelligence, with the use of CRISPR technology, we are now improving the way we design, proteins that we can now package them and utilize them for our own benefit. So, and I think, yeah, thank you.
Yes, sir. Thank you so much. Here is next question. If with the help of CRISPR we can edit or knock out genes, then why we cannot cure AIDS or any other disease which is incurable?
Well, if you remember, sickle cell is incurable, but it's not curable using CRISPR-Cas, right? So the same approach maybe people can apply to see whether you can cure or block HIV or other diseases, because it's based on, for example, HIV is the case of the receptor to which the virus needs to dock in, and this so what allows that interaction of the virus and the receptor. People have been developing tools or drugs-based, chemical-based approach in order to block the access of the virus so that they can terminate the progression or the disease that way, as using small chemical inhibitors or like the retroviral that people are using. So, but you can use this CRISPR technology maybe to modify and target how the receptor present itself in such a way that it is no longer able to. But that can be done in a way whereby you do not lose the functionality of the receptor, because the receptor naturally is supposed to be doing something like the so-called chemokine receptor, if you remember, 5, which is the one which HIV targets. So we have to maintain the functionality of the receptor, but we can find way of modifying something that can target where you can protect people from the virus having access to that. It's possible, but these are things that takes time. The ideas are there, but then it is only when you put them into practice, into trial, that you know whether you are able to achieve such success. So those diseases that are not curable at the moment, I'm sure with the advent of CRISPR technology, people will find a way, hopefully, of applying such technology to overcome some of the limitation. Even in terms of targeting the drug, for example, resistance, blocking, people develop HIV, a retroviral drug resistance. So what the mechanism that allows that? If we know the mechanism, then we can find a way of altering some of the structural entity within the body system or within the pathway in which this thing manifests itself, and find a way by which we can alter that, manipulate that, and so that it's no longer there or it's no longer available for it to manifest that will lead to such a resistance. So it's a time factor sort of thing. Ideas can be there, but then it is only when we put them into practice or trial that we find out whether we're able to do that or we are unable to do that. But as we are unable to do that also, we find a way of answering the question, why are we not unable to do that? And then in attempt to answer that question, then we probably may find a way how to do that. Thank you.
Thank you so much, sir. Here is the next questions. What are ethical concerns regarding CRISPR editing? And for today, how much advancement we have achieved in this technology, at which level it has been using as medical treatment?
Yeah, there are ethical issues. And as you know, with any experimentation or anything, nobody will allow you to go and have trial on people or anything. You must seek approval, but also at the same time, ethical consideration are given. But also equally, countries, the ethical issues are issues that are of international and national dimensions. So countries, through convention and other things, we may put a red line, say for example, because, for example, can you use CRISPR system in order to fundamentally change the human race? Okay, by certain manipulation or certain thing, which is possible. But then, are we ready for that based on ethical issues or ethical dimension? So it's still the same traditional ethical thing we need to ask ourselves. We need to address them. And as I say, there are ethical issues because there are probably certain no-go areas at the moment, just like with the issue of human cloning, because I'm not aware of any country that has approved anybody to clone a human being. But we have the enabling technology to clone human beings. There's no doubt about it. But then ethically, nobody is allowing that to happen except people may be doing it under the dark corner of their distance, but that is a risky thing to do, a very dangerous thing. And from research perspective, highly unethical to do that because whatever research we do has to be approved by ethical committees that allow us to carry out such the use or utilization of such technology. So they are concerned, I agree. And these ethical concerns are in a number of ramifications, either political, either economic, either religious, all these, as I say, the normal buzz points in terms of ethics will still be there to guide and control and regulate the use of CRISPR system. But also it's a decision for international convention, but also it's a decision for national interest because each nation may have its own policy. But then that policy must also align with what the international consensus or agreement is regarding the technology. Thank you.
Okay, sir. Thank you so much, guys. If you have any remaining questions, you can send me or Mr. Mutsa so we can take answers from the sir and share with you. And thank you so much,
Yes,
sir, for such an
no
insightful
problem.
session.
Yeah. Mhm.
And Mr. Ghulam Murza is here. Over to you, sir.
Thank you so much, sir, Professor Dr. Yusuf Deeni. I really thankful for your time. I really appreciate for being here. So at the next moment, we want to just inform you. about the report submission and report writing. Okay, we will
Okay.
discuss just right now. Okay, so be connected with the
Okay.
link
Thank you for
with the
having me
live.
then.
Yeah, thank you so much, dear
Yeah.
professor.
Yeah.
Yeah, you can now leave, and we
Yes. Thank
will
you.
share your email with the students. So if they have any question, they can mail you
Yeah.
their
No
questions.
problem.
Yeah,
Can do that. Yeah. No problem.
yeah. Thank you so much for
Thank you so much.
your time.
Yes. Thank you. Good night.
Good night.
Uh,
So the students, hello everyone. I hope you are enjoying good health and enjoying the session with a Nigerian professor. Actually, he's from Nigeria. He's all education, higher education, even MS, PhD, and postdoc in life sciences from UK universities. So he's in Nigeria just to serve his country. Uh, at the next moment, I just want to inform you about your report. Like this report is too much mandatory for certification process. First, you will submit your report at the end of that course in January. Okay. So you will prepare your report by as you take your classes. For example, you take 1 day class today, and you will take screenshot. You will proper report on your LinkedIn account, and then you will mention their organization as well. So organization, our social media teams will support you back on your LinkedIn account as well. So at the next moment, for example, attendance is mandatory for everyone. 90% attendance mandatory for certifications. So after reporting on LinkedIn your class, you will take a screenshot and you will put into your report, and you will write about your course, your first class with speaker name and each and every details about the class. So in this way, you will make and you will prepare your report of 8 classes with international speakers, and you may find the sample how you will make that report. We already shared in group. So I just want to inform you like one thing, we cannot uh, approach, you can say we cannot guide one by one to each single person. So we create that group for you people, so we can assist all of you in the form of group. But individually it's not impossible. It's so be connected with group and read the informations and instructions carefully, and then if you have any specific question even, then we will assist you, we will solve your problem as well. Okay. So the last thing, uh, for example, you come here, you invest your time. Time is important, and behind the scene, like behind the scene of that course, there are a lot of struggles of our teams of international collaborators to make that course happen, especially for you, especially for dedicated learners. So it is very important. You must have to take active part. You should have to enjoy that course, and it will helpful for you as well in future, hopefully. Thank you so much for everyone for being here, for joining class.
at the next moment you can yeah, leave the class, and see you in next class tomorrow. Soon we will share you a proper time of class. Best wishes, and Allah Hafiz.